Everybody. So as you noticed, we are here in Zoom and not in YouTube because we had a technical issue. So Mariana explained to you, we are going to be in Zoom today. So before we proceed with the presentation, I just want to make two very quick statements, which is the followings. So because we are in Zoom, we are going to try to upload then this presentation to YouTube. So please, I would suggest you to turn off your cameras and your microphones because otherwise when we put this in YouTube afterwards, there's going to be some problems if somebody doesn't want to appear, okay? So, and remember this also when you are doing the questions in the end. So if you don't want to appear in YouTube, you should always maintain your camera and your, at least the camera off, okay? So be careful with that. This is the first announcement. Then also, I will suggest that if you have questions, you can put your questions in the chat of the Zoom session. Although I really ask you not to get into dialogues in the Zoom chat. Okay, so if you want to put your question, just write it there and leave it. And at the end, I will see if we have time. Then I will ask you to talk. It's nicer than to just read your questions. But if we have time, then we do that. Otherwise, you can put your question there, but please don't get in the chat back and forth. Okay, just leave your question there. So having said that, it is my pleasure to have here in our Encontro Ciência Filipe de Sousa. Thank you very much for being here. I guess we are going to hear about bryophyte phylogeny. Filipe is a member of the COBIC team, but then today I believe he will do his presentation on a collaboration with Luís Catarino, and therefore I will pass the floor to Luís. Thank you. The floor is yours.
Good morning. I'll talk a little bit about Filipe de Sousa. Filipe de Sousa is graduated in biology at the University of Porto in 2005 and completed his PhD in evolutionary biology at the University of Gothenburg, Sweden in 2015. His main research interest is in the diversity and the evolution of plants, and this work has been mostly focused in the taxonomy and phylogeny of several genera of angiosperms, but also on the evolutionary relationships among the major terrestrial plant groups. Presently, Filipe is working with Otávio Paulo at the COBIC group of Center for Ecology, Evolution and Environmental Changes. He was invited by myself several months ago, and his presentation today is entitled "The monophyly of bryophytes is supported by nuclear and plastidial protein phylogenies." Thank you, and good presentation.
Thank you.
So thank you very much, Luís, for having invited me to do this presentation many months ago. Thank you to Professor Otávio Paulo for having me in his group. And now I will share my presentation. Here it is. Is the image okay?
Yes, I can see.
Yes,
okay.
It is okay.
So the title of my presentation is "The monophyly of bryophytes is supported by nuclear and plastidial protein phylogenies." And we'll see what this is all about in a minute. I am currently working at CE3C at the COBIC group, but I must say that this research I'm about to present was carried out at Centro de Ciências do Mar, which is a partner institution of the University of the Algarve. All the funding for the research was by FCT, Fundação para a Ciência e Tecnologia, and it was done under the supervision of Dr. Simon Cox at CCMR. It occurred, this research, between 2016 and 2019. So the broader question behind all the research I'm about to present is how did land plants first evolve? So how did diversification happen after the transition to land? So to place the land plants or embryophytes in a broader context, they belong in the green plant kingdom. And the green plant kingdom can be divided in two main groups. The chlorophytes, that include most of the marine algae, and the streptophytes. Within the streptophytes, we have the embryophytes or land plants, and a grade of several lineages that are often called the charophytes. These charophytes are very diverse morphologically. They can have more complex morphologies or just be unicellular filaments. For example, the group that is thought to be the sister lineage to land plants, the Zygnemateles, that includes Spirogyra, consists of simple filaments of one cell, for example. So the transition to land of the land plants is thought to have occurred in the late Silurian period. This was about 420 million years ago. In this illustration that I took from a recent paper from 2018, we can see that different lineages of green algae made independent adaptations to freshwater environments on land. But the definitive transition to terrestrial environments occurred only in the land plants, and it is thought to have been possible thanks to interaction of ancestral lineages with soil bacteria, and possibly involved even gene exchange. So what happened after the transition to land in the late Silurian? How did the lineages evolve? How did they diversify? And what characterized their morphological adaptations and their life cycle? This is what we tried to answer, because it was never an easy question to answer, as we will see. We can identify four main lineages within the embryophytes. Three of them are collectively called the bryophytes and include the hornworts. For example, genus Anthocerus belongs in this group. The liverworts, that includes genera such as Marcantia, and the mosses, for example, Sphagnum. The fourth lineage is the tracheophytes, which in its turn is divided in several other lineages: the ferns, the lycophytes, the gymnosperms, and the angiosperms. But it's the relationships among these four lineages that have not been easy to infer. And molecular evidence has actually supported contradictory hypotheses. For example, the most accepted hypothesis that we see in textbooks showed that the liverworts split from the remaining land plants. This was supported by, for example, chloroplast and mitochondrial molecular data. But the same data under other analyses instead placed the first split between mosses and the remaining land plants. Of course, that this then has implications on how we interpret, for example, morphological adaptations such as stomata and life cycles. Other scenarios placed mosses and liverworts forming a clade. This clade has been called clade setophyte. And we see that this conflict is not only between the genomic compartments, the nucleus, chloroplast, and mitochondria, but also between data types, meaning that analysis of nucleotide data often are in conflict with analysis of the corresponding protein data. More recently, thanks to next generation sequencing, people have been able to generate large data sets. An example is the data set published in 2014 by Wickett et al., which is a large RNA-seq data set. From this data set, two new hypotheses emerged as the most likely to explain the diversification of land plants. One of them places the hornworts instead as splitting first from the remaining land plants. And an equally supported hypothesis placed all three bryophyte lineages forming a monophyletic clade, meaning that the first split in land plants after terrestrialization was actually between bryophytes and vascular plants. This hypothesis has been supported not only by nuclear data, but also chloroplast data. The problem is that different analyses support one or the other hypothesis, and this has never been conclusive. So what could be the cause for so much conflict among these phylogenies based on different data types and genomic compartments? Well, regarding the nucleotide data, one cause for conflict is obviously saturation, because we are talking about highly divergent sequences from a very large geological scale of hundreds of millions of years. Nucleotide data is affected by saturation, which is basically a high rate of accumulation of substitutions at synonymous sites. The consequence of this is homoplasy and erroneous phylogenetic signal. A commonly used approach to mitigate this problem is the exclusion of third codon positions from nucleotide data. Of course, we're talking about coding sequence. Usually, to code for an amino acid, the third codon position can have any of the four bases. So by eliminating third codon positions, we get rid of most synonymous sites, and we can mitigate in part the effect of saturation. However, three amino acids allow for changes in the first codon position and even in the second codon position. These amino acids are leucine, serine, and arginine. So to mitigate the effect of saturation at first and second codon positions, because we can't get rid of them, we can use an approach called degenerate recoding. That means that changes at first and second codon positions can be replaced by a letter that will illustrate this change. For example, to code the amino acid arginine, when the third codon position has purines, A or G, the first codon can be an A or a C. So on the data matrix, when the codon for arginine has an A or a G in the third codon position, we can replace the first position by an M that represents A or C. So basically, we can transform the data set prior to the analysis to neutralize the effect of saturation at first and second codon positions. Another and more complicated cause of phylogenetic conflict in highly divergent sequence data is composition heterogeneity. This is basically a biased accumulation of specific nucleotides or amino acids that can result in the clustering of taxa with similar composition. Composition heterogeneity can occur among sites, for example, in a large concatenated data set, different sites can have different compositions, but it can also occur among lineages. So with time, different lineages will fixate or select certain nucleotides or amino acids and result in a variation of composition among lineages. So we have sites heterogeneity and tree heterogeneity. The problem is that commonly used models of nucleotide substitution in phylogenetic analysis programs do not model composition heterogeneity. They assume a single composition along the data and along the tree. So this can result in compositional attraction trees. And the classical example is here in bacteria. We can see a true phylogeny on the left, where three thermophilus bacteria appear, Thermus, Equifax, and Thermotoga. But often when inferring this phylogeny from molecular data, we get a compositional attraction tree where the thermophiles appear clustered. This can be possibly related to favoring of C and G in the data. So as we can see, it is necessary to model composition heterogeneity to avoid this problem of homoplasy and compositional attraction. So now I will present three studies, each focused on a different genomic compartment, starting with the nucleus, then chloroplast and mitochondria. So here is the citation to the first study, if anyone is interested. For this study, we selected a subset of the dataset that I mentioned earlier, Wickett et al., a large RNA-seq dataset. We selected from this dataset 100 genes and 26 taxa. Genes were chosen to minimize the effect of composition heterogeneity and choosing the shortest tree lengths. We sampled as outgroups charophyte lineages, including the sister group to land plants, the three bryophyte lineages, and the tracheophytes. And within the tracheophytes, we had representatives of the four lineages of tracheophytes: lycophytes, ferns, gymnosperms, and angiosperms. We analyzed nucleotide data with two strategies: the deletion of third codon positions and the degenerate recoding, to compare the two approaches. We used this in a maximum likelihood framework. We then analyzed protein data using both homogeneous or stationary models that do not model composition bias, and also with heterogeneous models, so to model composition heterogeneity. We also analyzed the protein data with the site heterogeneous model. I will make a parenthesis just to explain how we can apply the non-stationary models, so the tree-heterogeneous models. We use a program called P4, which is implemented in Python. So people not used to command line programs might find it a bit difficult, but if one starts with a pre-written script to run an analysis, it's not so difficult. Here is the web page, if anyone is interested. This is from the Natural History Museum in London, and it's Dr. Peter Foster that maintains the site, and he's very happy to answer questions. So to run an MCMC with a heterogeneous model, we have a script that points to the data set, transforms it into an object, a Python object. And here we have several priors to define the model applied, and several priors to define the MCMC itself. This program has the advantage of allowing for the extension of the analysis several times. So now to show some of the results, the analysis of the nucleotide data with simply the third codon positions removed placed the hornworts as sister to the remaining land plants. This is the result presented in Wickett's paper as one of the most likely scenarios. However, when we use the degenerate recoding that neutralizes the effect of saturation at first and second codon positions for arginine, leucine, and serine, we see that bryophytes emerge as a monophyletic group with very high support. This is bootstrap support, so it's quite high here. This is just to show how saturation at first and second codon positions in only 3 amino acids can have such a strong effect in a phylogeny and show completely contrasting scenarios. So we then analyzed the amino acid alignments corresponding to these nucleotide alignments. And we used Bayesian analysis on the program I showed, P4. So the protein data always recovers bryophytes as a monophyletic group. However, using a homogeneous model that ignores composition heterogeneity, the support, which here is a posterior probability, is quite low. So this does not allow to draw any firm conclusions. However, using the best fitting heterogeneous model in the program P4, we get the same tree with very high support for the monophyly of bryophytes. So in conclusion, using the heterogeneous models increases support for the monophyly of bryophytes, but also we applied a heterogeneous model that models heterogeneity among sites. This can be done in a program called Phylobase. And also here we get an increase in support for the monophyly of bryophytes. So we can see that these data are affected not only by tree heterogeneity, so composition heterogeneity among lineages, but also among sites. Unfortunately, it is not possible to combine the two in a single analysis, site heterogeneity and tree heterogeneity, not for the moment. But these analyses were conclusive regarding the monophyly of bryophytes, and we showed that the non-monophyly of bryophytes is driven by saturation at first and second codon positions in nucleotide data, and that support increases when modeling heterogeneity. We then decided to analyze a Chloroplast data set, and here is the reference to that study. This was a smaller data set consisting of 83 genes and 30 taxa, and the analysis of nucleotide data from this data set using a homogeneous model recovers mosses as sister to the remaining land plants. So a very different scenario here. When we analyze the nucleotide data with also a homogeneous model, but using the degenerate recoding that neutralizes the saturation at first and second codon positions, then this signal that places mosses as splitting first from the remaining land plants is lost. Instead, we get mosses and liverworts forming a clade that is not supported. So it's quite unresolved, but we can see that the signal that places mosses as sister to the remaining land plants in Chloroplast data is driven by saturation at first and second codon positions. Then we thought that even when we neutralize this effect of saturation, the synonymous sites may be subject to composition biases anyway. So we decided to analyze the degenerate data set with a heterogeneous model. And when we do that, the bryophytes emerge as a monophyletic group with full support. Also, when we analyze the protein data that should correspond to this, we get, again, full support for the monophyly of bryophytes. Although in the Chloroplast data there are some problems in the tracheophytes, possibly long branch attraction, we were able to conclude that Chloroplast data supports bryophytes as a monophyletic group. Finally, we had to look at the mitochondrial data set. Unfortunately, it was not as conclusive. It was a more complicated and smaller data set of 36 genes and 26 taxa. In this study, we used the ferns and the Zygnema talis, so the sister group to land plants, for the first time. And we got different results from the previous study on the mitochondria of land plants. So these data basically support mosses as the sister group to the remaining land plants, where nucleotide data is analyzed independently of the model used, either homogeneous or heterogeneous. The nucleotide data always places mosses as the sister group to land plants. The codon-degenerate nucleotide data instead shows the liverworts as the sister group to the remaining land plants under a homogeneous model. But when the heterogeneous model is used, then mosses and liverworts form the clade setophytes, and hornworts appear as sister to the remaining land plants. And the same result is obtained with amino acid data. So the tree with the best score of all the trees that we inferred placed hornworts as sister to the remaining land plants and recovered the clade setophytes that includes mosses and liverworts. Although this is not showing the monophyly of bryophytes, it is still more compatible with the monophyly of bryophytes than the previous study on the mitochondria. Also, I must say that although this was the tree with the best likelihood, other runs of the program did recover bryophytes as monophyletic. But when we have trees with different results, but one has a better likelihood, then that has to be chosen as the hypothesis. So in this case, we were able to assure that we recover the clade setophyte, but we could not conclude about the monophyly of the bryophytes or not. And we still think that the underlying phylogeny of the mitochondria is actually monophyletic bryophytes. But of course, it's a complicated data set. In any case, we could not reject monophyly of bryophytes very strongly with these data. So in conclusion, the analysis of nuclear and chloroplast data with better fitting models support the hypothesis that bryophytes are monophyletic. Data from all three genomes either support or fail to strongly reject the monophyly of bryophytes. We also showed that support for the non-monophyly of bryophytes is a consequence of saturation and composition heterogeneity. The implications of these studies allow us to reinterpret both morphological novelties that happened after the colonization of land by land plants, and also regarding the life cycles of these first plants. For example, we know that stomata are present in the tracheophytes and in the sporophytes of mosses and hornworts, but not in the liverworts. So when the phylogeny that placed the first split between liverworts and the remaining land plants was accepted, then the interpretation of stomata was very simple. They appeared after the split between liverworts and the rest. But if we consider that liverworts are nested within the bryophytes, then we have to consider that stomata are probably a simplesiomorphy among all land plants. So they were present in the ancestral embryophytes and was simply lost in the liverwort lineage. Also, the gametophytic life cycle was considered ancestral because bryophytes have a gametophyte dominant life cycle, whereas tracheophytes have a sporophytic dominant life cycle. When the phylogeny that placed tracheophytes as deriving from bryophytes was accepted, this reinforced the idea that the ancestral life cycle was actually gametophyte dominant. Although there was a different theory that said that the ancestral land plants could have had alternate generations that were isomorphic. So now we can perhaps consider this other hypothesis as a strong possibility that the first land plants have had an alternation of generations with an independent gametophyte and an independent sporophyte. This seems to be the case with certain fossil genera from the Rhynie-Chert flora. The Rhynie-Chert is a deposit from the early Ordovician in Great Britain. So the early Ordovician was just after the colonization of land. And certain genera display independent sporophytes and gametophytes. And although they are now considered stem tracheophytes, perhaps they actually have retained characteristics from the early embryophytes. Finally, regarding nomenclature, we recommend that the name Bryophyte is applied to all three bryophyte lineages, not just the mosses. And if a rank is chosen, then it should be a phylum with three classes: Anthoceratopsida, Marchantiopsida, and Bryopsida. And we consider this will be possibly an advantage when teaching botany, because bryophytes do share a lot of characteristics and are easy to interpret as a natural group. So this is all I had to say. Thank you very much for listening, and I'm open to questions now.
Okay, Filipe, thank you very much for this really nice presentation on using these so very modern tools to clarify bryophyte phylogeny. It was really interesting, and although I do not have any comments here in the chat, I would ask if anybody, because we have a little bit of time indeed, I would invite anybody that wants to comment or ask questions to do so, please. So you can just put your hands up or just turn on your micro and talk, please, if you wish.
Yeah, so I can start.
Yes, go ahead.
Okay, maybe if there's a comment, because I still need to reformulate my questions.
Okay. Okay, Vitor, so we'll wait a little bit for you. And then Manuela, please go ahead. Hello.
Okay. Good morning to everyone. It was a very interesting presentation. My congratulations. But when you talk about the monophyly of the group of the bryophytes, and we have data that points to the very early appearance of liverworts and in a meaning in between the mosses, which are now in the phylum, in the division bryophytes, and then Tocerota, more close to the first vascular plants. And also because there are some other morphological characters as the vascular transfer tubes that were found in some mosses, and also the connection between the gametophyte and the sporophyte in the Anthocerota. That transfer cells point to a closer relationships of this group to the first the group of the Poliosporangiophyta as Orneophyton and Aglaophyton. Did you consider some of those characters in your observations, in your research?
No, I'm actually not a paleobotanist, nor a bryologist. So I basically picked up this information of the Rhynie-Chert flora because I know it's a very heated debate that has been strongly supported by this hypothesis of the liverworts being the first lineage to diverge. But regarding the characters in hornworts, I mean, even if we accept this hypothesis of a split between bryophytes and tracheophytes, that's the polysporangiates would be the stem of the tracheophytes. If we follow the description of these of the Rhynie chert. They're called polysporangiates.
Yes.
But the characters in the hornworts would be easy to explain, because the hornworts are always the first to split from the other bryophytes. So even if we consider them a monophyletic group, because they are the first to split, perhaps they retained some of these characteristics that they share with the polysporangiates. So I don't think considering the bryophytes as a monophyletic group contradicts that. This is my opinion.
Okay, thank you. May I have another question very quickly?
Yes, please. Sure, go on.
In this group from Rhynie chert, there are evidences that some of them do not have any kind of vascular vessels. As instance, Cooksonia, that first was referred as having a kind of tracheophystes, very simple, but now it is published that it has no vascular vessels yet. So it seems that we're a kind of population mixture with several polysporangiate, with several sporangiates, but some of them already with some very primitive vessels and other with any kind of vessels. So it seems that in the evolution, there were times and that communities had several kind of representatives, bryophytes with this kind of group, that some representatives were more close to bryophytes, other more close to tracheophytes. So I think that some of them would have had more success and could establish to the vascular plants, other not. So it's still very tricky yet to have a clear idea of what could have been the evolution in these very ancient groups. So it's only my...
Yes, but I think what you said, in a way, I think it reinforces the idea that these fossil plants from the Rhynie chert cannot be assigned with so much confidence to the tracheophyte lineage, like you say, because there was a mixture of characters, some with a protovascular system and some not. Then perhaps these are just a group of stem embryophytes, and that the diversification happened afterwards. And this is what we are trying to say, that perhaps, for example, in terms of life cycles, the ancestral state would have been an alternate independent generations that then the sporophytes would have become dependent on the gametophyte in bryophytes,
Yes.
and the gametophyte dependent on the sporophyte in the tracheophytes.
Yes.
So yes,
very old
difficult.
theories, but the homologs and antithectic theory.
Yes, that's it.
Okay, thank you.
Thank you.
Thank you, Manuela. So I guess, Vitor, if you want, you can go ahead.
Yes. So very interesting. My question was more on the technical aspects of the phylogeny reconstruction, because I think it's very interesting to account for this heterogeneity, as you mentioned. And my question arises in the fact that for the mitochondria, you get a bit different picture, if I understood correctly, from the chloroplast and the nuclear genes. And I was wondering if the number of taxa and lineages that you include can somehow affect this heterogeneity. And I was just thinking if you can have some type of unbalanced, if you have many representatives of a given group that have for some reason more A and T, if that would affect and could explain why you get these different results with different...
Yes, I think so. And I think the ideal scenario is that you actually sample all the major lineages. Unfortunately, we're talking here about analyses that are rather heavy. Some of them took several months or even half a year, and sometimes you get to the end and it didn't go very well. So we have to keep rather small data sets. But like you say, a broader sampling, it is possible that it would solve the tree in a different way. But regarding the mitochondria, like I said, I mean, this is a Bayesian analysis, and sometimes it goes one way, sometimes it goes the other. The best scoring tree was the one I presented, but I did get other topologies, and I did get the monophyly of bryophytes. But I think possibly to get a final conclusion, I would have to do, like you say, different sampling with especially more bryophyte lineages. More hornworts would be important, but there are few full genomes for the hornworts, for example. And then with more lineages, perhaps the result would be different, and extending the analysis longer, I think.
Okay, but just to be sure I understood, but in the different data sets, my question was if the mitochondria could be different because you have a different set of lineages only in that data set. But if I understand correctly, you use the same taxa in all of them, or?
The taxa differed a little bit, but I mean, they represent the lineages. Usually we try to choose within each group the first lineage that diverged and a more derived lineage. So I don't think the problem lies there so much.
Okay.
I think it's more about the actual number of lineages, because the fewer lineages you have, the longer the branches, and it makes the phylogenetic inference a bit more uncertain and more difficult.
Okay. Okay, thanks.
Gabriela, posso pôr uma pergunta agora?
Sim, sim, claro. Go ahead, Otávio.
Então, so my question is also about why you have that different results in mitochondrial DNA, but if there could be an explanation related with the relative rates of evolution of the mitochondrial DNA genes versus the chloroplastidial genes and so on. And so just guessing. And the other question is that you try subsets of genes and say, instead of using a single data set with all genes, that you try different subsets and see if some genes have a more, say, content information that provides for one solution instead of the other solution.
Yes, unfortunately, while that is possible with the nuclear data set, with the mitochondria, because it's a small genome, I already used as many genes as I could by excluded genes that didn't have representatives of the outgroup or the tracheophytes, and I don't think. I don't think in the mitochondria we can do so many iterations of the data set. Regarding the rates, I think it plays a role. Yes, I did also consider a scenario in which the hornworts would have retained an earlier variant of the mitochondria, but I don't think it's very likely. I think it's just the data that is complicated. It's this combination of very high saturation that is very overwhelming in the mitochondria with the composition. And I think the solution here, like I said, would be to sample more mitochondrial genomes in the hornworts, because it's the complicated lineage, and maybe the other bryophyte lineages as well. I think that might be the solution, because the underlying phylogeny in the mitochondria is most likely compatible with the other two genomes, I think.
Okay, thank you.
Okay, thank you, Otávio and Filipe. So I don't see anybody raising the hands or putting anything in the chat. So you also have a comment here on Nagori Garcia, thanking you for the talk and saying that it was really interesting. Besides that, I would ask if anybody else wants to make a comment or a question, or maybe Luís Catarino, I don't know if you want to say anything, or Anybody else? If there are no more questions or comments, I guess there are no more questions and comments indeed. So I would like to finally thank again very much Filipe for this very interesting presentation. It also gave rise to some very interesting discussion, which is always very good. Thank you very much, Filipe. Thank you everybody for being here. I want to give a special thank you to Marta. Marta could not be here today for personal reasons. That is why I am here instead. But anyway, I want to thank her for organizing always this Encontro Ciências so nicely. And of course, I also want to give a special thank you for Mariana, which is always here in the backstage and that was able to quickly change our session from the YouTube to the Zoom. So then we are going to try to upload this Zoom session in our YouTube channel, okay, so that we have all these, you can also see later the presentation and the comments. So thank you very much, all of you, for being here. We are welcoming you to come back to our Encontro Ciência next week again, and please keep up with our conferences, okay? Thank you very much. Thank you, Filipe, again. Thank you, Luis, and all the participants. Bye-bye. Thank you.
Bye.